2012, she joined to Professor Graham Legroff's lab in New Zealand to continue working on protective immune response against helminths, but this time against hookworms. Currently, Dr. Tiffany is an immunology parasitology senior postdoctoral in Professor Harris' laboratory in Monash University, Australia. Dr. Tiffany has a strong interest in finding sustainable therapeutics against helminths, either a vaccine or chemotherapies. And next year, she will be opening her own laboratory in the Swiss Tropical and Public Health Institute. Congratulations, doctor. And she really loves worms, and you can find in her office a lot of stuff related to them, like plushies, 3D prints, and drawing of them. It is of great her love for worms that her 3-year-old kid knows what worms are. And please, again, if anybody has questions during this the talk, type them in the chat box, and I will read them at the end. So it's time to listen to you. Dr. Tiffany, the audience is yours.
Thank you for this lovely introduction and for the invitation for giving a presentation today. So today I would like to show you some of a story that we published a bit earlier this year in Host and Microbes that really focus on the importance for neutrophils. So apparently there is quite a neutrophil focus today with Neutrophils brasiliensis. So just to give one step back before Nepo, actually what we are trying to understand is how to treat the Hookworms disease. And Hookworms are still affecting about half a million people worldwide, and they are a leading cause of anemia and protein malnutrition. We do have treatment and we use mass drug administration that are at the moment efficient. However, there is no long-lasting protection against those parasites due to this protection. And we do know that resistance is already appearing. So there is really a need for new treatment against hookworm, and vaccination is seen as the most sustainable control strategy. However, to date, there is only one real vaccine candidate on the pipeline, and there is really a need for identifying new candidates. So really shortly to reintroduce Nepo model, I'm not going to give the full life cycle again, as you've heard it already. But what I would like to bring your attention to is the skin phase that I will mainly focus my talk on today. And why the skin phase? It's because Neutrophils brasiliensis, like the hookworm human Netosis, is actually a skin penetrating parasite. And actually that phase has not been really massively studied due to technical complexity of it. But we know that the parasite actually resides in the skin for about 13 hours. And during those 13 hours, there is something quite exciting that is happening. It's that the parasite actually transition from this free living stage to the parasite stage. And in particular, what it begins to do is releasing secretory products that we know have incredible modulatory potential. And the way we've been using to actually being able to study this phase in the skin is actually to fluorescently label this parasite. Here you can see a double labeling with the red that you can see is actually inside the parasite, and the green is the outside of the parasite, the schist. And using this labeling, you can actually follow the immune response that attack the parasite. And so really what I was interested in was understanding whether the secretory product could be a good candidate for vaccine development against those hookworms. And for that, we use the approach of using intradermal injection of those secretory product from the L3 stage and vaccinate. We have then production of antibody that block the function of ES in theory, and we wanted to know whether there was going to be any protection. And as you can see, indeed, we do have a decrease of the number of larvae that actually makes it to the lung after this vaccination. So it was quite encouraging. So now we wanted to understand, so what is the immune response happening in the skin? And for that, we use intravitreal imaging. And here what you can see is 2-photon microscopy. The larvae is in red. It's being intradermally injected in the ear of a mouse. And now you can beginning to see the cells, the lysine GFP positive cells that could be neutrophils swarming against these larvae. they actually swam really fast, about half an hour to an hour after infection. The larvae are completely covered up. So it was really quite exciting to us when we've seen that. But of course, because we are injecting directly with a needle, the neutrophil recruitment could just be the injury itself. So I actually wanted to know whether it was a parasitic response or whether it was just injury driven. And for that, I told you before that Netosis brasiliensis is a skin penetrating parasite. And here, if you can follow the parasite that I showed by the arrows, they actually enter the skin. So here you show that it is possible to have skin penetration, and we can follow it by imaging. However, this is a really unsuccessful event. Somehow it's only 8% of the parasites that enter the skin, probably because Netosis brasiliensis has been passaged for decades by subcutaneous injection, and so have actually lost this potential that is naturally happening with the hookworms. Nonetheless, there was actually enough entry of those parasites so that we can follow the immune response. And here you can see that once again, even earlier in the infection, we can see the neutrophil really around, or Tier1 positive cells around those larvae. And you can see that they really go around the schist, but later on, they can also attack the schist larvae. As you can see, a few of them actually are trapped around 11 hours post-infection. So really showing that this is a real phenomenon. It was not only the needle causing it. And so because we used either lysem or GA1 initially in our story, we didn't really know whether it was neutrophil or monocyte that were recruited at this site. So begin to use more specific staining here for monocyte or neutrophil. And what you can see is that actually both of the cell types are recruited. But what was quite striking was that actually monocytes are always further away from the larvae, and the neutrophil are the one that make contact with it. And we wanted to confirm what we've seen by imaging using in vitro settings. So just simple co-culture of the parasite with either purified monocyte or purified neutrophil. And once again, what we've seen is that without further activation, the neutrophil are actually quite good at directly binding to this larvae. So we were really excited and thinking, wow, okay, so what are neutrophils doing against helminths? Because you will not really think that a neutrophil will do anything against such a big pathogen. But actually, when we actually depleted either with LY6G or with GA1, we had exactly the same result. And we've seen that there was a delay in the migration of the larvae to the lungs, suggesting that maybe the hookworms larvae were able to sense the neutrophil and alter the development in response to this presence. And to confirm that it was exactly this sensing mechanism and change in development, we were to look at another developmental thing that happened in the skin, and it's the exschistment that I mentioned before. So the larvae enter with their schist here in green, and then the exschist, and then you can see them in pink. And so by using this strategy, you can quantify what's yellow when they're double colored. So basically the parasite is still in their schist, or what's only one color. And by quantifying, we were able to see that in absence of neutrophil, we actually had increased exschist parasites. So really showing that the behavior of the parasite has been modified by the presence or absence of those neutrophils. And not only does it modify their behavior, but it actually affects their survival. Indeed, if you look later on in the infection in the gut stage, you can see that in absence of neutrophil, you actually have an iron 1 burden. To finally confirm that neutrophil could actually kill the parasite, we went back to our in vitro system, co-culturing the L3 with the neutrophil. I have to say with an excess neutrophil, so that they could actually kill. And you can see that we could reach about 50% of deaths in vitro in 24 hours. So, all right, this is quite exciting. Neutrophil can indeed do something, but how do they do it? And at the time I was working on that project, the laboratory of Nicola was still in EPFL in Lausanne, in Switzerland. And Venizelos Papagenopoulos, an expert in neutrophil biology, came and gave a talk on this paper, where he actually showed that the neutrophil were able to sense the size of the microbes, and in his case was large hyphae. And when they sense really large pathogen, they enter by releasing nets. And so I thought, well, all pathogens are definitely large. Is it exactly what is happening in our case? And so to address whether nets were important in the phenomenon that we had described with the neutrophil, we actually took several approaches, either degrading the nets by DNase treatment or blocking their formation altogether by blocking PAT4 or neutrophil elastase. And you can see that in all cases, you have exactly the same result as what you have in absence of neutrophil. So this change in the behavior of the parasites to reach the lungs. And associated with that, we also had an increase in the worm burden in the intestine, showing that, yes, the neutrophil can kill, and they can kill in a netosis-dependent fashion. So at that point, I wanted to go back to the intravital imaging and being able to actually image those beautiful nets. However, when I've done the experiments, what I realized is, well, there is no nets. There's definitely neutrophil recruitment again around my live larvae, but absolutely no nets. And actually, Judy Allen and Venizelos Papagiannopoulos had published a paper in the lung that actually after neutrophil recruitment in the lung, there was no nets to be found either. So it was quite striking that I could show that the nets were doing something, but I could not see them. And so began to say that maybe the parasite had this evasion mechanism from the net, as many other pathogens have. So I used dead larvae that cannot produce the ES. And here you can see those beautiful nets appearing around the larvae. So to prove that it was indeed an evasion strategy contained within the ES fraction, we actually took those dead larvae once again and co-injected with the purified ES from the L3 stage. And what you can see is that you still have the neutrophil there, but you do not have the neutrophil extracellular traps anymore, really showing that the ES contains this evasion strategy. And in collaboration with the laboratory of Venizelos Papagiannopoulos, we were able to show that actually this activity that was blocking the net formation was actually degrading those nets. This is what you can see here. Increasing concentration of the ES was actually changing the lifetime of the net. So here it's nets induced by fungi, and then you can see that they are degraded over time, really showing that the function that we had that was doing the evasion is a DNase. Where further being able to show that it's actually a DNase II, which is quite uncommon. Other pathogens that are defending against nets use DNase I. But because we had published a few years earlier with the group of Alex Lucas, the secretome of Nipostrongylus brasiliensis, we knew that there was only 52 proteins in this L3-ES fraction. So the group of Alex helped us with that, and using bioinformatic tool have been able to actually find the DNase II in these proteins, and turns out that it is quite conserved throughout the strongylids order, really showing that potentially this DNase II could be a potent vaccine candidate against hookworm. And really to prove that it was the DNase II that was important, we've generated the recombinant and then generated blocking antibody against these DNase, and then went back to our in vitro system, and this time added the blocking the anti-DNase antibody. And when you can see that you can even increase further the killing made by the neutrophil, really showing that this is the DNase II that is the evasion strategy here. But as I told you, it was quite conserved. I was quite interested to know whether it was just a nipple phenomena, whether it was actually happening with hookworms as well. And so Alex Lucas' group once again sent us some Necator americanus larvae, and I've repeated the same experiment that I've done before with Nipo, showing that indeed the live Necator americanus were able to defend themselves against the nets with this reduction that you can see. It's quite easy to see in these images that are quite striking, that if they're not activated, so they don't release ES yet, you see those beautiful nets formation, while you have absolutely no nets formation at all when the parasite is able to defend himself. So, but what we realize as well is that not only can be good vaccine candidates, but we also have a DNase that is quite different from any other DNase shown so far that can actually degrade nets. And they can degrade any type of nets. It's just an example of PMA-induced nets, but I told you before that they were degrading also the fungi one, which means that this DNase could actually be used for treating inflammatory diseases, that are NETs mediated, such as lupus or here thrombotic microangiopathy. So really on that note, I would like to finish up this story showing that by looking at both sides, the host-parasite interaction, you can actually learn new mechanisms that could be harnessed for therapy against parasite. But at the same time, you can also learn this evasion strategy of the parasite that you could harness to treat inflammatory disease. And here was quite an interesting story where we show for the first time that neutrophils were actually swarming and directly binding to the larvae and entering netosis. But the parasite actually has its own response to this problem and then begin to release ES that can actually cleave those nets, thus permitting evasion of the parasite. So this DNAse II, we're quite excited, could be a really good vaccine candidate. There is probably many more protective mechanisms that are completely hidden by this evasion strategy and that we could discover by such an approach. And finally, there is a good chance that we can also uncover more of those immunomodulatory potential of ES using the skin phase. On that note, I would like to thank, of course, Nicola Harris' lab, where all the work has been done, and in particular, Dr. Matthew Moya, that have done most of the work with me, as well as fantastic honor student that helped finishing up that story in Monash University, and all the great collaborators that we had along the way that have really made this whole project possible. And I would like to thank you for your attention, and I would be happy to answer any questions.
Okay, thank you very much, Dr. Tiffany. Does anybody in the audience have a question? Well, I have some questions for you, please.
Yeah.
Have you considered that the stimuli that the larvae have to induce the secretion of nets could be a factor adhered to the larva or a soluble factor?
Yeah, so we know the DNAse II is soluble. So it's definitely secreted in those ES products. And we can find it in the soluble fraction. There is probably other things that are blocking the nets, because we do know that blocking the DNAse II is not as efficient as blocking netosis altogether. But in particular, we have identified in those ES protein that there is an anti-elastase molecule. So that could be something of interest to pursue.
Okay, and when you observe that in the presence of neutrophils, the larvae stay more time within the sheaths, but when you graph the results and quantify the number of larvae in the lung, you observe that there are fewer larvae that in the presence of neutrophils. So this is because the larvae are going away to the, running away into the lung, into the gut, or why?
Yeah, so it seems quite contradictory when you look at it at first glance, but in the end, what we think is happening is that they stay as long as possible inside the sheath when there is the neutrophil, because the sheath is probably protecting them against neutrophil killing. However, their speed of actually reaching the lung is different. So what they do is that when there is neutrophil attack, they stay long inside the sheath to protect themselves, and then they move on as quickly as possible to the lungs. So we don't really see any changes in their arrival in the gut. We only see the changes in the arrival time in the lungs, but it's probably just the speed to which they actually reach the bloodstream within the skin that is affected.
Okay, there is a question in the chat. Juan Inclán Rico says, excellent talk, Tiff. I wonder if the fact that neutrophil depletion that results in a delay of the worm migration from the skin suggests that the worm migration is more promoted by the host response rather than the worms continue with their life cycle.
Yeah, I agree. Most likely it's both of them, right? The life cycle, probably the worms have intrinsic control, but they definitely react to the presence of the immune system and adapt their their migration. It's been shown already for other parasites, for example, that filariae definitely change their development in presence of or absence of eosinophils. So it's something that is quite known, that the parasite can sense the immune system and change its development in response to it.
Okay. And Jaime Ocampo says, greetings from Mexico. And Christopher Moreno, microscopies were beautiful, just a question.
I think we also have a question from Alexandra Herens. It says,
yeah.
Yes.
Great talk. You saw less larvae in PID4 knockout mice in the lung. How was the larva number in the gut and also at a later time point in the lung?
So how were the larvae wet? Sorry.
How was in the PID4 knockout mice? How was the larva number in the gut and also at the later time point in the lung?
Yeah, so I have to say that we actually have not done later time point on those PAD4 mice. So this was what was asked by the reviewer to finish up the story, and we've just done the enough experiments in the lung stage early on to look, but it will be interesting to see. I would expect it to be quite similar to what the GR1 depletion was giving anyway.
Okay, and Christopher Moreno, did you look for any other extracellular producing cell, such as eosinophils or mast cells?
Yeah, so we are currently trying to look at eosinophil role in the skin phase and see whether actually they are doing also extracellular traps that could be participating. We've looked at the monocyte as well, because you've seen that they were recruited and they definitely do release extracellular traps as well in response to NIPO. But we think that the evasion strategy that we described here with the neutrophil will be functional against any other type of extracellular traps, given that it's just DNase based.
Okay, thank you very much. And Paula Licona says, do you know if these evasion mechanisms are conserved in other parasites, specifically targeting nets?
Yeah, so we looked at in broader Helminth's phylogeny whether we could find this DNAse II. The thing is, when you get out of the strongylids, the diversity of sequence begins to be way more important. And so we do not know. There is definitely DNase II, but there will be DNase II anyway, because this is important for the survival of cells. So we do not know whether they will have the same function or not. It will be really interesting. There is, in particular, for Trichinella spiralis, we found a sequence that was actually quite close. So it will be quite interesting to see whether those parasites also have the same evasion.
Okay. And I have another question, please. Do you know how long does it take to the production of nets? Because in your microscopy images, you observe that in the presence of neutrophils and larvae, when there are larvae, you observe that there is not nets around. But when the larvae are dead, you can observe the nets, but you can observe stained cells too. So how do you know that the nets are not being released by the cells?
Yeah, so here I showed 6 hours post-infection and the striking difference between the live and the dead. But actually, if you do earlier time points, more an hour or 3 hours, you have nets everywhere. So the live larvae will have nets surrounding them. The difference is that over time, those live larvae degrades the nets. So by 6 hours, you don't have anything anymore. While the dead larvae, they're formed around 1 hour and then they're sustained. So we have this whole kinetic, it's in the paper as well, if you want to see.
Okay, thank you very much. So we don't have any more questions. So we thank to Dr. Tiffany Bouchery for this great talk. And we will continue with the other